For this experiential learning assignment of statistics and decision making, our team is going to talk about Sir Ronald A. Fisher, the father of modern statistics, his contributions, developments, and discoveries in the field of statistics. Ronald Alamo Fisher, a pioneering statistician and geneticist, profoundly shaped the field of statistics through groundbreaking contributions. He revolutionized experimental design by introducing randomization and ANOVA, essential for unbiased and precise experiments. Sir Ronald Alamo Fisher was born on February 17, 1890, and passed away on July 29, 1962. Contributions: Design of agricultural experiments. Fisher focused on designing and analyzing agricultural experiments to improve crop yield and soil productivity. He introduced statistical rigor to the field by emphasizing randomized and replicated experiments. Statistical techniques. He developed and refined statistical techniques tailored to agricultural research. Fisher's development of the analysis of variance (ANOVA) method was particularly significant, enabling researchers to assess the impact of various factors on crop yield and soil quality. Application of mathematics to agriculture. Fisher's mathematical prowess was crucial in applying statistical principles to agricultural problems. His works laid the foundation for the use of statistical methods in agricultural research, paving the way for more precise and reliable experimentation. Statistical methodology. Maximum likelihood estimation (MLE). Fisher introduced MLE in the early 1920s as a method for estimating the parameters of a statistical model. The likelihood function. Fisher emphasized the more importance of the likelihood function, which represents the probability of observing the given data for different parameter values. Applications of MLE. Fisher's MLE has widespread applications across various fields such as economics, biology, engineering, and more. It is utilized in fitting distributions, estimating regression coefficients, modeling survival data, and in many other statistical analyses. Experimental design. Randomization. Fisher emphasized the necessity of random assignment of treatments of interventions to experimental units in controlled experiments. It helps mitigate the effects of unknown or uncontrollable variables that might otherwise bias the results. Statistical inference. Randomization strengthens the basis for making statistical inferences. It enables the use of probabilistic reasoning and statistical tests to assess the significance of treatment effects. Efficiency and precision. Randomization, when combined with replication, the proper statistical analysis leads to more efficient and precise estimates of treatment effects. Reducing bias. By randomly assigning treatments, Fisher introduced a method to reduce systematic biases and confounding variables that could affect the outcomes of an experiment.
ANOVA is known as the analysis of variance, which is a statistical method used to assess whether there are any statistically significant differences in the mean of three or more groups. It is a versatile and widely used technique that allows researchers to compare means and investigate the impact of categorical variables on a continuous outcome. There are two types of ANOVAs. The first one is one-way ANOVA and the second one is two-way ANOVA. The first one tells us about how it compares mean across three or more independent groups, whereas the second one examines the influence of two independent variables on the dependent variables. Key contributions of Sir Ronald A. Fisher. First one, introduction of the F distribution. Fisher introduced the F distribution, which is a probability distribution that arises in the context of ANOVA. The F statistics is calculated by dividing the variance between group means by the variance with groups. Partitioning of variance. Fisher emphasized the importance of partitioning the total variance in the data into different components, variance between the groups and variance within the groups. Third, the analysis of variance techniques. Fisher developed the ANOVA techniques as a way to test the null hypothesis that there are no significant differences among the mean of several data. Experimental design principles. Fisher contributes extended beyond just the development of ANOVA. He also played a crucial role in promoting the principles of experimental design. Fifth one, random block design. Fisher introduced the concept of random block design, a type of experimental design that helps us control for any source of variability other than the treatment being studied. Sixth one, practical applications. Fisher worked on ANOVA, had practical application in various fields, including the agriculture, biological, and medical. These are few general formulas used in ANOVA. The main formula is F equals to MST by MSE. F here denotes the ANOVA coefficient. Here is a problem shown about how we calculate the ANOVA coefficient, which is F. Here they have given the number of animals, the average domestic animals, and the standard deviation. By using this data, we can summarize that the ANOVA coefficient is equal to MST by MSE. Applications of ANOVA in statistical analysis. There are mainly 8 applications. The first one is comparing means in multiple groups, quality control in manufacturing, medical research, analysis of survey data, analysis of education data, analysis of variance in sports performance, economics and finance, and analysis of genetic data. Genetics and evolution. Genetics is a branch of science which deals with the studies of genes, genetic variations, and heredity in organisms. Evolution is a process of heredity changes in the population of organisms over a several period of time. Fisher's impact on genetics and evolutionary biology development. The 8 main impacts are the quantitative genetics, genetics variability and natural selection, fundamental theorem of natural selection, genetic correlation, population genetics, experimental design in genetics, statistical methods, and legacy and recognition.
Hello, my name is Avikshith and I am here to discuss the next few slides on Sir Ronald A Fisher. We will have a brief discussion of controversies and criticisms faced by Fisher. I will name the few categorized ones: statistical rigidity, personal disputes, and controversial interpretation of results, and resistance to Bayesian statistics, and limited recognition of collaborators. Now, in statistical rigidity, Fisher was known for his strong opinions and at times a rigid approach to statistical methodology. Some critics argued that his insistence on certain techniques, statistical techniques, limited the methodology and exploration of alternative methods, which hindered the development of more flexible approaches. Personal disputes. Fisher engaged in several professional and personal disputes with other prominent statisticians of his time, most notably Carl Pearson and Jerzy Neyman. These disputes often revolved around differing views on statistical methodology. Controversial interpretations of results. Fisher interpretations of statistical results were not always without controversy. Some critics argued that his strong emphasis on p values and hypothesis testing led to a narrow and at times misleading interpretation of research findings. Resistance to Bayesian statistics. No. Fisher was known for his skepticism towards Bayesian statistics, a school of thought that uses probability theory to model uncertainty. His resistance to Bayesian methods sparked debates within the statistical community. Limited recognition of collaborators. Fisher's tendency to take sole credit for collaborative work and downplay the contributions of his colleagues such as Egon Pearson has been a point of criticism. This behavior has strained relationships and led to some dissatisfaction within the scientific community. We'll move on to the next slide. Legacy of Sir Ronald A Fisher. His legacy can be mainly categorized into theoretical impact, impactful books, and statistical hypothesis testing. In theoretical impact, we already have discussed the importance of maximum likelihood estimation and analysis of variance. They have become fundamental in statistical theory. Impactful books. In Fisher's life, he has written 8 books, which have been extremely useful to the statistical community. Statistical hypothesis testing. He has created so many important things such as formulating null hypothesis, proposing alternative calculate test statistics, assessing p-values against alpha, and making decisions based on predetermined significance level. We'll move on to the next slide. We'll discuss some things about Fisher's significant testing and Fisher's price index. Firstly, we'll go to Fisher's significant testing. It involves formulating hypothesis, calculating a test statistic, accessing p-value, and making decisions based on predetermined significance levels. Fisher's price index calculates the average change in prices of a fixed basket of goods, providing a measure of inflation. We'll move on to the next slide. Fisher's exact test. Well, to give the definition, Fisher's exact test is a statistical method used to determine if there is a significant association between two categorical variables by calculating the probability of obtaining observed frequencies or more extreme ones in a contingency table. But simply put, Fisher's exact test helps us figure out if two things are related, looking at how likely it is to get the observed results just by chance. This is the table. This is how to get the p-value. We'll move on to the next slide. Fisher's publications. These are some of the impactful books that I previously talked about. I have named some and showed the images of some. The design of experiments, the genetical theory of natural selection, statistical methods, and scientific inference. Now, Fisher had a major impact in agriculture. These are some agricultural applications of Fisher's findings. This is a question of how Fisher's findings can be used. Question. Agricultural researchers conducted an experiment with 3 different pesticides to control a pest. Can Fisher's ANOVA provide evidence of significant difference in pest control effectiveness? Answer is statistical analysis. Apply ANOVA to test whether there are significant differences in the mean effectiveness scores of 3 pesticides. If the p-value is below the chosen significance level, we reject the null hypothesis of equal effectiveness. Question. A farmer wants to determine if the application of a new fertilizer increases crop yield compared to the standard fertilizer. How would Fisher's method guide the statistical analysis of the yields? Answer. Statistical analysis. Conduct a 2-sample t-test to compare the yields of 2 fertilizer groups. If the p-value is below the chosen significant level, it provides evidence to reject the null hypothesis. No difference in mean yields. Let's move on to some genetic applications. Question. In genetic association study, investigating the presence of a specific allele with a disease, researchers collected genotype data. How can Fisher's exact test be used in this context? Answer. Statistical analysis. Apply Fisher's exact test to create a contingency table comparing the observed and expected counts of the allele in individuals with and without the disease. A low p-value indicates association. Question. Researchers studying a plant genetics wants to assess whether there is a significant difference in the frequency of certain trait between the two populations. How can Fisher's methods be applied? Answer. Statistical analysis. Utilize Fisher's exact test to compare the observed frequencies of the trait in the two populations. A low p-value suggests a significant difference in trait frequencies. These are some applications in the agricultural field and the genetic field, in both which Fisher worked for. Lastly, we can conclude that Fisher left a great legacy as the father of statisticians, leaving behind genetic applications and agricultural applications to the world of statistics. Thank you.